Okay, have you ever really stopped to think about one of the most incredible magic tricks happening in the universe? It's happening inside your body, right now, every single second. We're talking about how life packs an absolutely staggering amount of information into a space so small it's basically impossible to imagine. We're talking about DNA.
Let's start with a number. 2 meters. That's about 6.5 feet.
Now get this, that is the length of the DNA that's packed inside just one of your cells. I mean, a single microscopic cell. If you could actually pull it out and stretch it, it would literally be taller than you are.
So that brings us to the big question, right? How in the world does all of that genetic information cram itself into a microscopic nucleus without becoming a complete and utter tangled mess? Seriously, it's like trying to stuff a mile of spaghetti into a tiny salt shaker. It just shouldn't work. So, how does the body solve this 2-meter-long problem? Well, it turns out our cells have a whole series of absolutely brilliant solutions. Let's start unpacking this elegant system they use to manage what should be a data storage nightmare.
All right, the first hero of our story is a group of proteins called histones. The easiest way to think of them is as these tiny, positively charged spools. Now, your DNA happens to carry a negative charge. So, what happens? It's just simple chemistry. Opposites attract. The DNA is just naturally drawn to these histone spools. It's a beautifully simple and elegant solution. So the DNA wraps itself tightly around these histone spools, and it creates this structure that scientists have this perfect name for, beads on a string. And each one of those little beads, which is basically a bundle of DNA wrapped around some histones, is called a nucleosome. And just this first step, this one trick, shortens the DNA's length by about 7 times. But, oh, it doesn't stop there. The beads on a string thing is just level 1. It's the beginning of a whole hierarchy of compaction. Those nucleosomes then start to coil up into an even thicker fiber. Then, that fiber gets folded and looped even more, creating the super condensed structure that we call chromatin. So you've got wrapping, on top of coiling, on top of folding. So, we've managed to pack our 2 meters of DNA down into this tiny microscopic space. Problem solved, right? Well, not quite. See, this ultimate storage solution actually creates a brand new and pretty tricky challenge. Think about it. If your entire genetic instruction manual is locked away, vacuum-packed into this super compact form, how do your cells actually read it? How does the machinery that needs to access your genes get to the right page when the book is slammed shut and locked in a vault? And the cell's solution is just so clever, it's dynamic. It doesn't pack everything away with the same tightness. The chromatin can actually exist in 2 different states. First, you've got euchromatin, which is kind of loosely packed. It's open for business. The genes here are totally accessible and can be read. And then you have heterochromatin. This stuff is packed super tight. It's closed for storage, and the genes inside are completely silent. Okay, so how does the cell decide which parts are open and which are closed? It's not random. It uses something called the histone code. And this is amazing. It's basically a whole system of chemical tags that get attached to the tails of those histone spools we were talking about earlier. These tags are like little signals, telling the cell's machinery, hey, open this part up, or nope, lock this one down tight. And 2 of the most important words in this code are acetylation and methylation. When the cell adds an acetyl group, that's acetylation, it usually loosens up the chromatin, making the genes way easier to read. You can think of it like a big green go signal. Methylation, on the other hand, is a little more complex. Depending on where that methyl group goes, it can either tighten or loosen the chromatin. It's more like biochemical punctuation, giving more specific instructions. So the cell has this incredible dynamic filing system. It can pack its DNA away but still get access when it needs to. But you know what? There is still one more huge problem we haven't solved. The very act of using the DNA, copying it, reading it, that creates its own massive physical challenges. I want you to imagine trying to unzip a rope that's made of 2 strands and is thousands of feet long. As you start to pull the strands apart in one spot, what happens to the rest of the rope? It's going to get twisted up into a tangled, knotted mess, right? Well, the exact same thing happens with our DNA. When the cell tries to read or copy it, it creates this intense twisting force, leading to these things called supercoils and knots that would just be catastrophic if you left them there. So to solve this problem, the cell calls in another set of hero enzymes, topoisomerases. These things are the master untanglers of the cell. Their job is to relieve all that mechanical stress, and the way they do it is just wild. They literally perform a kind of molecular surgery.
They find a tangled spot, they temporarily cut one or both strands of the DNA, let the tangled part unwind and pass through, and then, and this is the important part, they perfectly stitch the DNA back together.
I mean, this process is so mind-blowingly precise and elegant that topoisomerases have earned this awesome nickname, the molecular mathematicians of the cell. Because that's what they're doing. They're seeing and solving these complex physical topological problems in real time.
But here's the thing. This incredible biological process, this essential act of cutting and rejoining DNA, isn't just some fascinating biological trivia. It's actually become a major target for modern medicine.
Yeah, by understanding exactly how these molecular mathematicians do their job, scientists have figured out how to hack the system to create some incredibly powerful drugs that are used every single day.
And here's the genius of it. In a normal, healthy cell, the topoisomerase makes its cut, lets things unwind, and crucially, rejoins the DNA. The whole thing is temporary. But certain cancer drugs, which are called topoisomerase poisons, do something sneaky. They let the enzyme make the cut, but then the drug jumps in and blocks it from completing that final critical step. It prevents the rejoining.
The drug basically turns the cell's own helpful enzyme into a weapon against itself. Those unrepaired breaks in the DNA, they're catastrophic. They're a death sentence for the cell. And this is especially deadly for cells that are dividing really fast, like cancer cells, because they're constantly copying their DNA and rely heavily on these enzymes to get the job done. By poisoning the process, the drugs essentially force those cancer cells to self-destruct.
So from a simple chemical attraction to a complex histone code, all the way to molecular machines performing surgery on our genome, the solutions our cells have come up with for packing DNA are just as elegant as they are essential for our survival. When you really look at this whole intricate system, you see how our cells are solving what should be impossible physical and logistical problems every single second of our lives. It really makes you wonder, doesn't it? What other elegant, brilliant solutions are still hiding in our DNA, just waiting for us to discover them?